So thank you for being here. This is going to be a very interesting session. We have two speakers, and the focus of this plenary session is going to be on perceptual integration and disintegration. And of course, these are really key topics in thinking about states and contents of consciousness. So our first speaker comes to us from UCLA, from the Department of Psychology and some other departments as well. This is her first time here at the Tucson meeting. We thank her for coming, so please join me in welcoming Dr. Ladan Shams.
Thank you very much for having me. It's really exciting to be here. I'm going to talk about the easy problem of consciousness, the sensory integration. And I hope that I will convince you that the easy problem is not all that easy. For our perception scientists, at least we find this a very computationally complex problem. In our daily lives, frequently we come across objects and events that produce sensory stimulation in more than one sensory modality. For example, when someone walks towards us, we can see them. We can probably hear their footsteps. If they're wearing a cologne, we can smell them. When we hold an object, for example, an apple, we can see the apple. We can feel the apple. And these sensory inputs are processed in largely separate neural pathways. The visual stimulus is processed in the retina, goes through optic nerve to thalamus and the visual cortex. The auditory stimuli are processed in the cochlea to brainstem, auditory pathway, and so on. And yet, our experience of objects and events is not a collection of separate sensations. It's a unified and coherent and monolithic experience. This experiential unity of consciousness shows that the merging or the binding of the different sensory signals happens prior to conscious awareness. So we perceive a 50-gram red apple as opposed to perceiving 50-gramness or redness separately. But there's more to multisensory processing than just binding the different sensory inputs. There are many properties of objects that provide information through different sensory modalities. For example, the size of the apple, the shape of the apple, the texture of the apple. These information are conveyed through both visual and haptic modalities. And the combination of the estimates that are provided by the different sensory modalities provides a better estimate about each of those variables, perceptual variables. And each of these estimates that are provided from the different sensory modalities are not perfect. They're noisy, they're unreliable. And we have found that combination of the different estimates provides an improvement in precision, in accuracy, and also in processing time of the sensory stimuli. But as I said, the estimates are noisy, and the sensory stimuli are processed in largely separate pathways, and therefore they are corrupted by independent noise processes. And because of this, there is discrepancy between the estimates that are provided to the brain about the same object property, like the size of the apple. Vision may tell us something, somatosensory input may tell us something else. And for the brain to be able to perceive a coherent object, this conflict somehow has to be resolved. And the source of noise can be external, like fog or dim lighting in a room like this, or dirt on the hand, and a number of other external factors. But most of the noise, we believe, is inside our heads because of the variability in neural firing, which we cannot really explain and we characterize as noise. So the noise is independent in different modalities, and that means that discrepancy or conflict between the different senses is the rule rather than the exception. So the brain has to somehow solve this conflict, and it has to determine which of these estimates are more reliable and how to combine them to come up with a better estimate, a more reliable and more accurate and more precise estimate. And this is a computationally non-trivial problem, and that's one of the problems that the brain or perceptual system has to solve all the time. Now, beyond the natural discrepancy that is that involves in processing of the sensory signals, the studies, classic studies of multisensory integration have created artificial conflict between the signals by pitting one modality against another to gain insight into what modality dominates in any given situation. And these classic studies of multisensory integration have all shown results that suggest that vision is the dominant sensory modality in humans. One example of this is the ventriloquist illusion, where we perceive the sound, in this case, for example, the voice of an individual to originate from the mouth of the puppet as opposed to the actual position of the sound, which is the puppeteer. So vision, the visual information captures the perceived location of sound. And this has been known for a long time, since 1970s, and yet the exact mechanisms were not clear until recently. And it's not a very obscure or contrived situation. It's an illusion that we experience all the time these days when, for example, we watch TV or we go to the movie theater. Every time that there is any discrepancy between the exact position of the sound and the visual stimuli, like the actors or the faces on the screen, this is, and we don't notice it, this is the illusion we are experiencing. A very similar illusion happens when we combine or when we pit the visual information against proprioceptive information. For example, if an individual comes to the lab and we have them wear a prism that shifts the image drastically to the right, so they position their hand on one side of the table and the image is shifted to the right, and we ask them where is the hand located, they report perceiving the hand in a location which is much closer to the image of the hand rather than the actual position of the hand. And this is known as visual capture, again showing that vision dominates another modality. This also happens in speech perception. If we pair the voice of a syllable, for example, ba, with lip movements associated with another syllable, then we tend to perceive a third syllable, which shows, again, the influence of visual information on auditory perception. These interactions are not limited to perceiving the events in the world. They also happen with respect to the perception of our own bodies, or our own body parts. And a very well-known example of that is rubber hand illusion. And here, the subject sits at a table, and we hide the hand, or for example, the left arm in this case, and we present a fake arm, a rubber hand, on the table. And the experimenter strokes the fake finger synchronously with the real finger. And after a few times of stroking, the subject experiences the rubber hand to belong to their body. So they start owning a foreign object as part of their body. And this has been known for a long time, and there have been many studies of this. It's a very freaky illusion. A lot of people get really freaked out by it. Another illusion that, again, pits visual information against another visual modality has been known for a really long time. It was first reported in 1891 and has been studied extensively over decades. is the size-weight illusion. And here we have two objects that have the same shape, like two cubes, and they look like they're made of the same material, and they have the same exact weight, and yet people perceive the smaller object as being heavier if they lift them at the same time or sequentially. So all of these have made scientists and lay people alike to believe that in humans, vision is the dominant modality. But if you look closely, we find that this is really not the case. We discovered an illusion that I'm going to demonstrate to you here. And for this illusion to work well, you need to fixate on that red dot. So that's the fixation point. And I'm going to present a stimulus on the left side, in the lower left side of the screen, if I can, on the lower left and also on the lower right. And while you keep your eyes fixated on the red dot, try and pay attention and see if you notice a difference between the two stimuli that are presented in the bottom of the screen. So here we go. Oops, sorry, somehow the sound is not working well. Try again. Okay, this is supposed to be one beep, so let's see if I can get it to work. Nope. Okay, so try this one. Okay, so for some reason the sound is not working here, but let me try one more time. Let's try our luck. Okay, this was better, and now another time this side. Okay, how many people notice the difference between what was presented in the left and right? Raise your hands. Okay, so now I'm going to repeat the same exact thing, but I'm going to turn off the sound, and I'm going to play the same QuickTime videos again. Okay, now how many people felt that these two stimuli were identical? Okay, so when there is no sound, it's very obvious that the two stimuli, the black disks, flash once in both cases. But when there is sound, it becomes really difficult to perceive that they are identical. In fact, if the sound worked well, you would perceive one flash on the left side and two flashes on the right side. And this is because the disk on the left was paired with one beep, the disk on the right was paired with two beeps. So what is happening is that when we pair a brief visual stimulus with two sounds, it is often perceived as two or more flashes. This is known as sound-induced flash illusion, and it's a really robust illusion because we can manipulate the shape, size, location of the visual stimulus. We can change the sound. We can even tell the subjects that there is only one flash, and still people perceive multiple flashes. And even if you have seen this a million times like me, the illusion still works for you. So it is somewhat immune to top-down cognitive influences, and it's a very robust perceptual illusion. And the influences, so this is a very nice demonstration that vision is not always the dominant modality. Other modalities can significantly and radically alter what we see. But even in a task that is generally known to be a visual task, like spatial localization, that typically vision dominates, like in ventricular illusion, if we manipulate, if we change the stimuli such that the visual image is very impoverished and sound is very strong, then we can reverse even this illusion, and then sound captures the location of the visual stimulus. And this was demonstrated by Allais and Burr a few years ago. So which modality dominates at any given task, any given situation, depends really on the task and depends on the specific stimuli. And this is now quite well established in perception community. But even though all of these phenomena that I presented seem to be very diverse, involve different tasks, different kinds of modalities, they all have one thing in common, and that is that in none of these cases, the observer is aware of a conflict between the senses. And we have no access to the unisensory information. When we perceive the double flash, we cannot tell that, oh, this was one flash and 2 beeps, or the original or the unisensory stimulus was different from the bisensory experience. We have no access to the unisensory information anymore. But is this the case only when the signals of the different senses get completely fused? Or is this a more general phenomenon? To look at this more closely, I'm going to use the spatial localization task as an example because it's easier to notice the different phenomena. And the fusion is something that we already discussed. For example, when the 2 stimuli, the sound and the visual stimulus, are presented at the same location, of course, like if the speaker and the screen are at the same location, we perceive it as one object, and we fuse the two sensory signals. If the discrepancy is small, the same thing happens, and that's what we call ventriloquist illusion. If the discrepancy between the senses is very large, then we don't typically combine them at all. They're segregated. But if the discrepancy is moderate, then sometimes for some subjects, what happens is what we call partial integration. The 2 percepts get shifted towards each other, but they don't get completely converged. And this shows that even when the subjects perceive 2 independent objects, and even when they perceive a conflict between the senses, they're not aware of the interaction between the senses. So in general, we can say that the cross-modal interactions, whenever they happen prior to conscious awareness. Now, the research over the last couple of decades in a number of different laboratories using a number of different techniques, the research that has looked at this phenomenology of cross-modal interactions, the neural mechanisms of it, the anatomy of the brain, all of these have provided converging evidence that cross-modal interactions are ubiquitous, and they happen at multiple stages of processing in the perceptual pathway, and they start from very early stages of processing. We showed that, for example, the sound-induced flash illusion involves modulation of activity in V1, primary visual cortex, which was believed previously to be completely unisensory, or a sensory-specific area in the brain. And other laboratories have shown different things in other sensory modalities. And some recent results also show that interactions can also happen really early on in the auditory pathway at the level of cochlea. How the activity is affected, the activity in cochlea is affected by the direction of gaze, for example. So all of these are interesting, but everything I have told you so far is related to when we have one object or one event producing sensation in multiple sensory modalities. And this almost never happens in the world, in that we are almost never exposed to just one object or event in our environment. In the real world, we are exposed to a number of different sources of sensory stimulation, and the brain has to process tens of different sensations all at the same time. Even if we go to a really quiet environment like a library or walk in the woods, there are tens of different sensations across the various sensory modalities that are processed in the brain. So, for example, a woman having a quiet dinner with her family, her brain processes a number of sensations, such as the image and the voice of her husband who's talking, maybe the cologne of the husband, the feel and the image and the sound of the utensils she's manipulating, the feel and taste and the sound of the food she's chewing, the smell and the image of the salsa on the table, and so on and so forth. So there is a melange of all these neural signals that are being processed in the brain, and none of these signals comes labeled with where they came from, what the source was. And it's the job of the brain to figure that out. The brain has to go from this mishmash of neural signals to something like this, where the causal structure is worked out, and we can tell that, oh, this image and this sound came from the same object, that feel and that odor and that sound came from another object, and so on. And this is the problem we refer to as causal inference. Causal inference has been exclusively studied up until recently in the context of cognition, problem solving, reasoning, and things of that nature, and had been really neglected in the context of perceptual processing. But I argue that it's a much bigger problem for perception than it is for cognition, because this problem has to be solved all the time, at every given moment that we receive any sensory input. And it is extremely complex because it is computationally intractable. We have no idea how the brain can go from something like this to something like this. It is computationally intractable, and we have no model of how it may be done. So again, demonstration of the easy problem of consciousness is really not all that easy. But to make it easy on ourselves, we can simplify this into a really artificial, simplistic scenario, where instead of having tens of sensory signals, we are only processing 2 sensory signals. And this is what we have done to make this more tractable for ourselves. And even in this very unnatural kind of situation, the problem of causal inference persists. The brain has to determine whether the 2 signals were caused by the same object or independent objects. So at any given moment, the perceptual system is encountered with 2 important problems. One is the problem of causal inference, which signals were caused by the same object, which signals come from different objects. And then for those signals that are estimated to have the same cause, the brain has to determine how to integrate them, which one is more trustworthy, which one is less, and which direction they should be combined, and so on. And that's the problem of sensory integration that I already mentioned, which is also a non-trivial problem. We proposed a Bayesian causal inference model that can account for both of, or can solve both of these problems, the causal inference problem and integration problem, in a coherent and unified fashion. And it's really simple. It considers 2 causal structures, a common cause and independent cause. And these are 2 hypotheses, or 2 models, or 2 prior expectations. And the model solves or computes the probability of each of these hypotheses using Bayesian inference. And once the probability of, for example, common cause is calculated, then the estimate of what is going on out there in the world can be estimated, again, using Bayes' rule. And as an example, if, for example, the task that we are performing is spatial localization, we want to know where the sound is coming from, then the optimal estimate of the location of sound turns out to be a really simple and elegant formula, which is a weighted average of 2 other estimates, optimal estimates. The optimal estimate of the location, assuming that the 2 signals have a common cause, and the optimal estimate of location, assuming that they have independent causes. And each of these estimates is weighed by the respective probability of the causal structure. So this weighted average, you could probably already see how this would result in or give rise to partial integration. Because even if you perceive that the 2 things have different causes, there is still an influence from the other modality on the estimate of the object property. And this causal inference model can also be thought of as a competitive prior model where we have different priors telling us that these signals have a common cause, and prior that says that they have independent causes. And these priors, these expectations, compete with each other to explain the sensory input. Okay, so comparative prior is another formulation of the same exact model. So can this model, it's simple and it's nice, but can it really explain how the human perceptual system works in various conditions? We have tested it for all of the classic multisensory phenomena that I mentioned. For example, for ventriloquist illusion, we presented subjects with sounds and simple visual stimuli, either at the same location or different locations, either presented in a unisensory fashion or bisensory, and we asked them to localize these stimuli, and we observed a spectrum of phenomena ranging from fusion to partial integration to segregation. And this figure shows the responses of the subjects. Each of these plots is data from one of the sensory conditions, the combination of audio and visual, and the solid lines represent the observer's responses, the human data. The broken lines represent the model fits. The model has 4 free parameters and accounts for 97% of the variance in the data consisting of 1,200 some data points. So the model really does a very good job of explaining the data for the entire range from segregation to partial integration to complete fusion. But in our study, we didn't ask subjects to tell us whether they perceive one unified object or independent objects. In another study by Mark Wallace and his colleagues, they did ask that. And their data is shown here in black, and the model predictions are shown in red. Again, the model can account for their data very well also. So this model can account for spatial perception. How about temporal? We use the sound-induced flash illusion as an example of a temporal task because what's happening is really a temporal numerosity judgment. And we presented a variable number of flashes and beeps, and we asked subjects to report how many pulsations they see in each they perceive in each modality. Again, the solid lines represent subject data, the broken lines represent the model fits, and the model, same exact model, 4 free parameters, accounts for the data very well in this task as well. The nice thing about this paradigm is that it can be nicely extended to more than 2 modalities, and that's what we did. We introduced somatic-sensory stimulation. We presented variable number of flashes, beeps, and taps on the finger, and we asked subjects to report what they perceive in each modality. This is the data. We had unisensory, bisensory, and trisensory conditions. And what we found was that there are a number of different interactions, and the sound-induced flash illusion that I demonstrated earlier, there's really nothing special about that. We found that every time there's a discrepancy between 2 or 3 modalities, some illusion happens, some interaction happens. So these interactions are really the rule rather than the exception. And we extended the model to take into account 3 sensory signals, and the model, again, with only 3 free parameters, accounts for 95% of the variance in the data, accounting for all of these varied interactions. So these are perceptual phenomena that estimate what's going on in the world. How about something which is, that has to do with the perception of self? To test that, we looked at rubber hand illusion. And here, this is a bit more complicated because it involves not only 3 modalities, vision, proprioception, and touch, but also it involves both spatial processing and temporal processing. And the model was simulated and predicted that when the stroking of the hand is synchronous, then subjects should perceive a lot of illusion, when the stroking is asynchronous, there should be almost no illusion, which is consistent with what has been reported in the literature many times before. But what the model predicted was that even if there is no touching, no stroking of the finger, sometimes there should be some illusion. There should be a moderate degree of illusion. And this had not been reported before. So we tested for that experimentally. And we found that indeed 75% of subjects report ownership of the rubber hand even when there is no stroking. And we used another experiment, we used GSR as an objective measure of ownership, and we found the same thing. So, okay, so not only, and the rubber hand illusion study was really intriguing for me because what it says is that the brain doesn't take anything for granted. We think that, we're born with our bodies, and therefore it should be a no-brainer. It should be hardwired that this is my arm and that's not part of my body. But the brain doesn't take any of that for granted, and it is always busy calculating what belongs to our bodies and what doesn't belong to our bodies. And the same calculations that apply to the external world also govern the perception of our own bodies. I am out of time, so I'm not going to get into the next illusion, which is my favorite, size-weight illusion. We looked at it. It was the biggest challenge to the model because it's considered to be an anti-Bayesian phenomenon, and our model is Bayesian. So we tested, we replicated the illusion, we extended the model, and the model provides a compelling account for this illusion, even though it is generally considered to be an anti-Bayesian phenomenon. And the McGurk illusion, we have also some data, unpublished data, showing that is governed by the same computations. So overall, we can say that the Bayesian causal inference model seems to govern, seems to account for a number of diverse phenomena involving different combinations of modalities, different perceptual domains, the world, the outside world, the internal world, and therefore seems to be the computation that explains or is recycled by the nervous system for a number of different tasks. The idea is not novel. It was proposed back in 11th century by a brilliant scientist by the name Alhazen, but now we have the data supporting this idea. We have a MATLAB toolbox of the model for those of you interested in playing around with it. So now, in conclusion, causal inference, even though had been neglected in the context of perceptual processing, is a key component of perceptual processing. And Bayesian causal inference seems to be a unifying account of multisensory perception. And Bayesian causal inference seems to govern not only the perception of the world, but also the perception of ourselves. And cross-modal interactions ranging from segregation to partial integration to full integration, they seem to happen prior to conscious awareness, and they are ubiquitous. They happen from very early on in the sensory pathway, and they're not unique to humans or even primates, and therefore it's really not all that surprising that they don't require conscious awareness. Thank you so much.
Thank you for that wonderful talk. Actually, we have a short session, so we're going to have our next speaker up, and then we'll have a panel. So it is a great pleasure to welcome Professor Tony Hudetz to the stage. Dr. Hudetz is a professor of anesthesiology and neuroscience at the University of Michigan, also the scientific director of the Center for Consciousness Science. He's going to talk about the other side of this question, which is disintegration. Dr. Hudetz.
Thank you very much, Dr. Mashur. Perfect. Yes, so you heard my title, and the title says Altered States of Consciousness, but I will limit the talk to anesthesia effect. And in a way, this will be, yes, a little connection problem. Good? Yes, thank you. In a way, this will be a continuation of the previous section, sort of a fifth lecture on anesthetics. And I'd like to, because this session is on integration and disintegration, so let me start with a couple of general comments. So first of all, integration defined as a coordination or combination of processes in a nervous system. And we heard about sensory integration, feature binding, multimodal integration in particular, wonderful examples, but we could also talk about sensory motor integration, cognitive integration. And of course, integration can be spatial and temporal, and it could involve local connectivity or local interactions and distant interactions, and could be temporal. In particular, temporal binding is a sort of integration. And we could also talk about information integration in various contexts. IIT is one of those, and in a nutshell, what that would mean that the whole is more informative than the sum of its parts. And so my focus is on disintegration, which I would define as a failure of integration. And the hypothesis I will be exploring is that unconsciousness can result from disintegration of information processed in the brain. So I'm going to start with an older experiments that we did in the early 2000s. At the time, graduate student and later postdoc Olga Ilmes and I did, in which we did experiments in rats with chronically implanted electrodes. You see in the upper left, electrodes were placed in various parts of the cortex, in frontal cortex, parietal cortex, temporal cortex, and various segments of the visual cortex. And we used coaxial concentric electrodes, and I just mentioned that it was important to localize, to better localize gamma activity. At the time, there was a surge of interest in gamma oscillations based on the work of Wolf-Singer and in case of Perception, Talon, Baudry, and others. So we started looking at gamma oscillations. And so we used that actually first rest, but then later with sensory stimulation, applying a light flash in one or two eyes of the rat with different means, stroboscopic illumination, and later with LEDs. And we saw that this simple stimulus that activates, sort of primes the visual processing across the entire cortex. If you look at the upper right panel, there was a very structured oscillatory response in the gamma range. These are gamma filtered, 30 to 60 hertz in this case. Signals in primary visual cortex V1 that propagated into parietal cortex, PTA, and further into prefrontal cortex. And when we looked at the coherence of these sensory evoked responses in the gamma range on the lower left, we found that indeed, in this case, isoflurane was suppressing the gamma coherence between the anterior and the posterior side and suggested that there was some sort of disconnection between these regions. However, we also noted that the reduction, and look at the dark black bars, the reduction occurred very early at a very shallow anesthetic concentration, while we know that rats lose consciousness, at least based on our surrogate behavioral index, the writing reflex, lose at approximately at 0.8% halothane or isoflurane, which is the same. And so this reduction was too soon. It may have accounted for memory change, but not really for consciousness. But then we went on and looked at gamma oscillations a different way. We took a wavelet transform of the gamma oscillations and calculated transfer entropy, which is a directed measure of interaction between regions, and that resulted in that lower right figure, which you have seen a couple of times at this conference already, expressing that what anesthesia really did near the dose where rats lose their writing reflex, presumably their consciousness, was a weakening of the top-down feedback connectivity. And this result was further corroborated by results on the evoked response in visual cortex itself. And here you see on the very left, and actually these are 4 different neurons, so top-down, and shows the firing response of the neurons. And what the light flash induces is a complex oscillatory response. You can see the waves, hopefully, from where you're sitting. But this oscillatory response is converted very quickly at the shallow level of anesthetics into a much more simplified response. And about the time, and that's the right column, by the time the rats go unconscious, the secondary wave is completely gone. And this is, of course, another indirect indication that the later integrative processes that occur, perhaps through feedback from higher regions, are suppressed preferentially by the anesthetics. So these results, together with other results obtained in Giulio Tononi's lab in Madison, Wisconsin, where they used transcranial magnetic stimulation, together with our results, were then interpreted in the context of information integration theory and resulted in this position paper, actually exactly 10 years ago, in which we described that the hypothesis that there were essentially 2 ways information disintegration could occur in the brain. And one was a disruption of the causal interaction between brain regions or neurons, so the level has not been defined by the theory exactly. Either a disruption or a reduction in the repertoire or diversity of brain states that the brain could access over time. And you wonder, if you haven't read the paper, you wonder about the dice. So there's a metaphor that we use to explain that. And what you see, the green globe is a multi-phase dice. And each of the face represents a brain state. And again, this is a metaphor. But you can imagine when we roll the dice, the faces follow in a probabilistic order of the dice rolls, so there is some interaction, some causal order. But if we take the faces away, in other words, we remove the interactions, that mean we disrupted the causal order. And the second example below shows that if the faces of the dice would be very similar, or in an extreme case would be all the same, like a single form, which would mean all brain states are equal, then this would be also a very uninformative situation. And so it would entail to a reduced information. So now this theory is nice. It leaves unanswered at what level of integration is important for consciousness, what level of disintegration, I mean organization level, would disintegration be causally important for losing consciousness. And that's what's on this slide. Question, we know that integration occurs in the brain at multiple occurs vertically as well as horizontally in the cortex, or hierarchically, if you like, across various regions. So it's a logical question to ask whether disintegration occurs perhaps at molecular, maybe microtubular, molecular level, synaptic level, local circuit level, regional level, whole brain, where is it? Also, the temporal scale, which you heard about, at what temporal scale does disintegration occur? So at the large scale or macro scale, the concept is really very simple. And what this graph shows is one of my early drawings. There are probably much nicer ones out there now. But it shows that the concept that during consciousness, the local modality-dependent processors, which could be associated with brain regions, in this case the yellow color regions, are connected in some way, exchange information with the integrating hubs, which is the pinkish color hubs, and this whole system processes information. But what happens under anesthesia, in the case of at the level of unconsciousness, that these subsystems become somehow disconnected. also between the integrating hubs and perhaps between the integrating hubs and the modality-specific processors. And then one more thing to mention, there's also a paradoxical case that happens in very deep anesthesia. We call this burst suppression, when the EEG becomes intermittently active and inactive. So bursts alternate with isoelectricity, and that's the right, that's panel C, when the system actually becomes hyper-connected. But it's easy to see that this connectivity, again, is information poor. If all the neurons and all the regions would fire exactly at the same time, there wouldn't be much meaningful information in the system. So what's important for the brain at large scale, I like to emphasize, is selective connectivity. Total connectivity would have very little information. No connectivity would have very little information. But selective connectivity is a candidate. Macroscale. So after this, we became more interested in what happens at the neuronal level. So I'm going to talk about microscale disintegration here. And this is an experiment, again, done in rat. And what you see on the upper left is a microelectrode array. And this microelectrode array has 64 recording sites, meaning that we can record from at least 64 locations in the brain, and this is inserted into the cerebral cortex, in our case, visual cortex in this experiment. And you can see the electrode spans all major layers of the cortex, 1 through 6. And I'm going to show you a brief movie. And this is what it, after processing the signals, looks like when neurons fire without any stimulation. So the hot colors mean very high firing intensity, and the blue color goes to low or 0. So there's an ongoing, very rapid and dynamic firing, even in the absence of stimulating the cortex. Visual cortex is active even in the dark, without seeing anything. It's always ongoing. Now, what happens in unconsciousness? As Stuart mentioned earlier, in the unconscious brain, there's still plenty of activity, but there is something peculiarly different in the unconscious state, and it's probably easy to see by eye that the activity is repeatedly disrupted. There are temporal gaps when activity all over this piece of visual cortex takes a break. And we quantify this. It's easy to quantify, group the neurons firings. If you look at the diagram below, every black mark is actually a time point of neurons firing, and the little red brackets indicate when neurons were grouped together into firing state and high firing state and low firing state. Neuroscience often mentions these as, or refers to these as up states and down states. And then we can do this, and the top row is under deep anesthesia, and the bottom row is awake. And you can see in the awake there is almost continuous firing, but in the anesthetized state, so the sedated, very deep to light, if you go top to down, then becomes, changes the pattern is changing, the temporal pattern. So now we're interested in disintegration. So what we want to look at is the correlation of these up states with each other. So this graph shows how correlated these high neuronal firing states are at each location with its surrounding location. So what the panel on the very left shows is in a conscious animal, neurons that are in the middle layers, and this is especially layer 4, and it's surrounding maybe bottom of layer 3 and layer 5. So it's mainly the input layer of the cortex where thalamic fibers terminate, are fairly well correlated. So there is correlation in neuronal firing in this layer. But as soon as we add a little bit of anesthetic, in this case 2%, this is desflurane, inhaled anesthetic, it just produces a very slight sedation. Then this correlation focus on the middle layers is disrupted, and the correlation spreads out. And if you increase the anesthetic further to 4%, which is the third column or third panel, then you see already a suppression, and at 6%, when the animals go unconscious, it's an almost complete suppression of the coordination of neuronal firing. So this is an example of disintegration. And I'm just going to throw this in as well. Another more direct measure, how do neurons communicate with each other, can be obtained with cross-correlating the spike times of neurons. And if we see a significant high peak on the very left, look at the panel on the left top, see a high peak that suggests there is a monosynaptic excitatory interaction between neurons. You see a trough, it's an inhibitory interaction, and the fact of whether you look at an inhibitory or excitatory neuron can be further verified based on a waveform of spiking, which is on the low panels, and their autocorrelation. But the main point I'd like to say is the upper right panel. What do anesthetics do to these pairwise neuronal synaptic communications? Is what we see is a substantial and significant reduction in the excitatory signaling. And I think this ties well with what Bruce McIver said about the suppression of excitatory transmission with inhalational anesthetics. However, the inhibitory connections seem to be unchanged or nearly unchanged. Now, I also like to get closer to integration. And so this is the third, and actually the fourth, example of microscale disintegration. And in order to say something more concrete about information disintegration, we calculate a property that's actually called integration. It also runs under other names, multi-information or total correlation. But basically what expresses is how much interacting. It expresses the amount of information associated with the neurons interacting with each other. Actually, to be more precise, we don't know whether that may be partially due to a common drive. The more specific, if we want it to be exact, it specifies the information associated with the correlations in the system. And this is calculated without going into too much detail. One takes the spike raster on the upper left, which is just the time points of the spikes, and we binarize it. So 1 means there is a spike, 0 is no spike. And the formula expresses a difference in the entropies of the individual spike trains versus the population. And the essential thing is that this quantity is 0 if the neurons don't interact, and it's some non-zero if they do. And so what do we see with anesthetics? It's on lower right. And again, here we use desflurane. And as we increase the anesthetic concentration from 0, fully awake, to approximately 6%, which is where the rats presumably lose their consciousness based on the writing reflex, we see an obvious reduction in this integration quantity. And perhaps importantly, or more importantly, the steepest drop in this integration occurs exactly in the range where we transition from a deep sedation, but still probably conscious at 4%, to where they completely lose writing reflex at 6%. And this, by the way, points to the importance of dose-dependent studies. It is important to know what exactly happens at the critical point where animals or humans lose or regain writing reflex. So this relatively sharp transition that I'm showing between 4 and 6% actually speaks to an older, but it's still interesting question. What is the transition like of going from conscious to unconscious, between conscious and unconscious state? Is it that if neuronal activity is changed by any means, do consciousness levels follow gradually, such that we apply anesthetic in an increasing dose? Does it happen like we would turn off the light with a dimmer switch? Or does it happen much more like an on-off switch, and we go through a very sudden, dramatic reduction? So I think this result gives a hint. This is, of course, an average result from many experiments. Hints that there might be some abruptness in the transition and are not completely graded as some of the earlier results showed, but there is a critical, perhaps critical transition. A further question was that we asked whether we can actually reintegrate the neuronal system. And in this experiment, we tried just that. And the way we approached that is we placed a stimulating electrode into the pons, to be exact, in the nucleus pontis oralis, which is known to be a center in the brainstem in the pons that gives rise to ascending activating pathways to the cortex. So it serves as mostly glutamatergic. It is a center that maintains or even activates the brain, the cortical activity. And we recorded in secondary visual cortex and parietal association cortex with more candidate for integrative function. And what we saw is if you just look at on the right panel, the left graphs, the very tall dark blue column shows how that it was a very excessive increase in integration. As soon as we turned on the stimulation to brainstem, integration went back up to very, very high levels. And we were also able to show that this kind of brain stimulation not only increases integration at rest, but it also increases the response to visual stimulation. And I know it's a very complex graph, but if you just look at the right side in V2 and compare the orange bar with the dark red bar, there is a small increase. So this before flash and after flash. But after brainstem stimulation, and now you compare the gray bar with the green bar, there is a much larger increase. So in other words, when you activate the cortex from ascending pathways, you can also enhance the integration quality. Yep. I have a few more minutes to talk about another scale. So this is mesoscale integration. So in mesoscale, I'm talking about local field potentials, for example. So with the very same electrode I used to record spikes, the same electrode on the left, we can, of course, from every location get field signals, and these would be local field potentials would reflect 1,000, probably 10,000 to 100,000 neurons. So it's a more global signal, but it's still a relatively micro signal compared to large scale. And here's the movie of what you see in the field potentials. And so the red is the positive and blue is the negative. The scale is normalized, so the units don't matter too much. But it is apparent there is an ongoing dynamic change in the field pattern. And this movie was prepared at 1 millisecond time resolution, so I slowed it down quite a bit for you to see. So it's a very rapid, constant formation of clusters, of potential clusters that change constantly. In the lower left is just a few examples of clusters. Now these potential clusters form and stay for a short time, and they dissolve and move to the next one. So therefore they're called metastable. And the question was that we had is how does anesthetic affect this dynamics? And one way to analyze this or quantify it is you just measure the time, how long these metastable potential configurations exist, and when they resolve, what is their duration? We call that a dwell time. And on the graph on the right shows that the letters mean wake. So as we go from wakefulness to light anesthesia to moderate to deep anesthesia, the dwell time increases at least twofold. So basically what this means is that these field potential configurations are logged in much longer into their state. And this is equivalent to state that over time, the repertoire or the diversity of brain states that the brain will access is reduced. That just during the same time, not enough, not that many states the brain can access. We can see this at a very different, jumping back to macroscale, at a different scale, and now you're going to see fMRI data, and this is again obtained from the rat. It's a resting state blood oxygen level dependent activity. Oops, sorry. It's supposed to play. Yes, it plays. And so what you see is bold activity in rest in a mildly, lightly sedated rat. In order to keep the rat motionless in the MRI scanner, we have to apply a small dose of anesthetic. And the very bright colors, as they form very dynamically, reflect clusters of co-activation. So at this level, we can also assess the question of repertoire. And on the right, by the way, just sequences of brain networks as they form in time, just to illustrate what they look like. But the results are on this slide. So on the left side is a measure of the temporal variance of these activation clusters. A temporal variance is an index of the repertoire of brain states, in this case defined at the MRI level. And what is very evident that compared to the conscious state, the state repertoire of these activity clusters is largely, and by the way, statistically significantly reduced. Another measure on the same data, we used algorithmic complexity. So not just how many states are, but how complex these states are. And this can be evaluated, for example, with Lamp-Ziv complexity. And the result that was published there shows again that in all animals tested so far, in the 6 animals, is a very clear drop in the complexity of these brain states. So I have one more experiment to talk about, and that is about dynamic causal interactions. And this is a work that's ongoing. It's in progress. Publication is in preparation, but I'd like to show you anyway. And the reason we go in this direction, because everything we have looked at so far was either steady-state connectivity, or it was pairwise. So we looked at neuronal interactions pairwise. But what we really want to do is when we record from a large number of neurons, in this case, in this one actually 74 neurons, how do neurons interact as a system? So we use the multivariate Markovian model, which is with a sliding window. And the sliding window, that means 4 seconds. So we evaluate the model at 4 seconds, and shift. And what this model is attempting to do, the multivariate Markov model, is it attempts to predict the firing of one neuron from the firing of all other neurons at the previous time step. And that means this is a causal measure now of the interactions of neurons. And you can do this for the entire system, predict the firing of every neuron from every other neuron, solve this equation system, and get of course, use statistical tests to determine the significance of these interactions. And I'll show you another movie. But before I click the button, so just to orient you, so on the horizontal axis is just a number of neurons. There are no labels, but 74 neurons. And the vertical axis are the very same neurons. But the difference is this is the state of the neuron at the previous time step, which is actually 4 milliseconds. And why is 4 milliseconds? Because we want to look at monosynaptic interactions. So just like before, synaptic transmission occurs between 2 and 4 milliseconds. So by limiting the time step to 4 milliseconds, you can guarantee that more or less monosynaptic interaction. So here's the movie. And what you see now, again, these are not neurons firing, but these are the interactions between the neurons, the temporal causal interactions that between all neurons. And it's obvious that there is a very dynamic, ongoing pattern. So 2 neurons talk to each other for a moment, the next moment they talk to somebody else, and they're driven by 2, 3, 4 other neurons, but then this changes. And changes at every 4 seconds in the lower right is just another depiction of this. It's a still figure of 10 seconds worth that's 100 seconds. 100 second worth of interaction. So on the vertical axis are the neurons, and every color line means that one neuron was driving another neuron. And then, of course, the neurons are numbered from top top down. So the final result from this, I see I have a few minutes, 1 minute, less than 1 minute. If you summate all these interactions, you get a dynamic interaction plot on the top in the conscious animal and one on the just below in the unconscious animal. And we average everything across time, then you get the lower graph. And what this shows is the total number of dynamic interactions as a function of anesthetic. We again use the S-plane. And then again, this graph seems to show a somewhat abrupt, or at least sigmoidal, transition near loss of consciousness. It starts a little bit earlier than the integrated information measure, but again, this is work in progress. So if I'm allowed, I'm just going to summarize. And so what I have shown you is different examples of anesthetic-induced disintegration that occurs in various spatial temporal scales. Disintegration is seen in large-scale disconnection and in the temporal fragmentation of spontaneous ongoing activity. The transmission probability of neuronal spikes at excitatory synapses is reduced by anesthetics, whereas the inhibitories are unaffected, at least by volatile anesthetic. And the information associated with the interaction of neuronal population is also reduced. And finally, the state-dependent change in integration seems relatively abrupt, at least sigmoidal. Percepts suggest a gentle on-off switch. Future investigations are many, but they probably should focus on the regional specificity of these changes. We only looked at visual and parietal. We need to look at prefrontal and many other regions, and also the cellular mechanisms of these measures of disintegration in neuronal systems. And I thank to my collaborators, and most of the work I showed was done by former grad students and later postdocs at MCW, but also several collaborators from University of Michigan already. I appreciate the help from all. Thank you.
Thank you very much. 2 wonderful talks, and Dr. Shams is coming up, and we can have some questions. I'm actually going to abuse my privilege and ask the first question because I think it's an important point that we should get out early on. So Dr. Shams, you talked about this integration being outside of conscious awareness, multisensory integration, but do you believe that it is an essential prerequisite for conscious awareness? And maybe just to have a little bit of dialogue with this question of integration and disintegration. And there's a microphone right here for you.
We are aware of the environment even when we don't integrate the signals and we perceive independent objects. But that doesn't mean that if we should integrate the signals, if they do have a common source, and we fail to integrate under conditions that Dr. Hudetz, for example, discussed. That I have no comments about that. I could easily see that if integration fails in conditions that it should take place, it could disrupt consciousness.
Thank you.
Hello.
And everybody keep questions and comments brief.
Yeah, this is for Dr. Shams. How much do you think your results reflect the fact that the brain is a prediction machine, if they do?
This is a very good question. The predictive coding models have become very successful in accounting for a number of perceptual phenomena. And our model, by taking the prior expectations into account, in some sense, it is a predictive coding mechanism that tries to predict what is going on out there in the world. So I think that's a very pervasive feature of perceptual processing, but predicting what is going to happen and what is out there.
Thank you.
Actually, I'm going to go to this side again just because we have a little bit of asymmetry. Sure. Please.
Thanks. So I'm interested in kind of zoning in on the relationship between...
Could you start over and speak a little bit closer? It's hard to hear you.
So My question has to do with the relationship between the multisensory integration processes and then a self. And so it seems that from the causal inference story, the self is used as a sort of causal object to bind some sensory signals, the diverse sensory signals, to that causal object. But then on the other hand, it's also suggested that the multisensory integrations underlie the building of an object of a self. So they sort of inform the building. So there's almost a chicken and egg. Does this multisensory integration underlie the self? And then does the self help multisensory integration? And I'm just wondering if your models or if your thoughts on sort of perhaps a bootstrapping process between a self object and multisensory integration. Yep.
So I'm not sure if I completely understand the question, but what we find is that the brain tries to find what is causing what sensory signals, and the sensory signals involve also signals we receive from about our bodies that include the image of our body, for perception of our body, the sensation of touch, etc. If all of these senses are congruent and they're telling us the same thing, then we tend to integrate them and we tend to perceive them as being caused by our bodies, ourself, and we own this source of sensation as ourselves. So that's what we find in rubber hand illusion, and there are other body illusions that have been discovered recently that are very much along the same line. For example, the causal inference model can also account for the out-of-body experience that has been reported in a lot of different experiments.
Yep. So
I'm sorry, we're going to have to move on. We have many other people who want to ask questions. Over here, sir.
Yes, this is for Tony. You mentioned the nucleus pontus oralis and the ascending arousal system and so on. Is it your sense that anesthesia turns down the brain, and perhaps that turns down the pontus oralis, and then if you activate the pontus oralis from where it may have been turned down, it then turns up the brain again? Is it something like that?
Yes, so now I have to add that obviously pontus oralis is only one of many ascending pathways. And, and in fact, the fact that we had any success at all is quite remarkable, because I would think to completely awaken an animal would require a number of pathways. But yes, I think that in this case, the assumption was the primary target of the anesthetic is cortical. But this can be overcome by ascending activation. Not saying that anesthetics do not affect the subcortical pathways. It's lower arousal from that end as well. So it's probably everywhere in multiple targets, but still you can overcome the depression, the cortical depression, by stimulating ascending.
And over here, sir.
Thank you. My question is about causal inference for Professor Shams. I had the experience recently as a trained musician listening to two equally sized men singing a song together, singing the same exact words throughout the whole song, but singing a harmony of each other. And it was actually quite difficult to tell who was singing what from where I was sitting. And by paying close attention to the discrepancies occasionally between what I could see in their lips and what I heard, I could pick out which was which, but it was very difficult. And my question is, why is it so difficult to concentrate on the enjoyment of the experience before my brain can figure out what's really going on and who's singing which part? Does that question make sense?
So it interfered with your enjoyment because you couldn't figure out who was producing the
the which
sound?
is basically a meaningless piece of information, but it was very important.
Drink more wine.
I'm glad that you raised this question. This is a very important topic that has been studied very little. We call this perceptual pleasure. We have started recently in my lab to study that. And the previous studies of perceptual pleasure have focused on individual senses. Nobody has looked at the relationship between the senses. So that's what we're starting to do, to see how integration and congruency between the different senses, sensory signals, influences our perceptual pleasure. I'm not going to give you a punchline that our hypothesis, because we haven't published it yet. We don't have definitive results yet, but I can say that it is not unlikely that whenever there's incongruence or some conflict in our, whenever the perceptual system has a hard time converging information, it is not pleasant. We find it unpleasant. But unfortunately, I can't tell more because we haven't studied it in detail.
Thank you.
So we have just about 5 minutes left, so very brief and concise questions and responses. Over here, please.
This is for Dr. Hudetz. You had shown a slide where you specified that unconscious states were associated with either a complete disconnection amongst neural elements or a hyperconnection, whereas consciousness was associated with a selective connection. If you look at the early complexity measures that Tononi and others developed in the 90s, it follows a non-monotonic function where at random states of a system, it's low, ordered states of a system is low, but in between, complexity is high. And then integration increases as you go from random to highly ordered. So do you think that what you showed there, this relationship between selectivity in the middle versus disconnection and hyperconnection, maps on to such a complexity relationship?
Yes, the answer is yes. Actually, we have looked at another measure that Tononi calls interaction complexity. We didn't look at phi itself because it's impossible to compute it for 100 neurons. Maybe for 10 you can do it. But complexity is a simpler measure, and that's exactly indeed we found. With interaction complexity, you have an optimum peak, and so when you get into burst suppression very deep, you have less complexity, and also you can get to the other side of the curve. So this is true. I did not show those data. I didn't have that many of them. But yes, with interaction complexity, you see this kind of inverted U from these data.
Thank you.
Hi, so also in the last lecture, you asked the question about the transition between conscious and unconscious. Is it moderate or more like a phase transition? So I didn't see, like the answer. What do you think about it? Is it a phase or moderate? And is it the same if it will be the opposite? I mean, when you go from unconscious to conscious, will it be the same, do you think?
I think that, just speculatively, there should be two elements of this. There are processes in the brain that change gradually under the effect of any pharmacological agent. But then, as a system, as an integrated system, state transitions can occur much more rapidly. So there's an interplay. I think based on our data, I cannot strictly say that there is a totally abrupt transition in the form of something like was suggested by computer simulation studies like Jamie Slay and others predicted that. But we're measuring here, and every measurement has some error. And so lots of assumptions and averaging over animals and all that. So just the fact that the transition curve is not a gradient totally linearly changing. The fact that we have much more of a sigmoid than a linear relationship suggests that there is indeed some kind of state transition, and we might be able to delineate it further to make it even more abrupt. And that's been a quest for our quest to find something that's some phenomena that changes more discretely than just simply dose dependently. And we're kind of happy that we found at least a sigmoidal transition.
Thank you.
Jan?
Thank you both for the wonderful talks. I have a quick question on the rubber hand illusion. You showed this really dramatic difference when they synchronize the stroking on the rubber hand and the real hand, and they get this illusion. But if you stroke asynchronized, they are basically has no illusion. But if you don't have stroke, then you have 80% of illusion. So my question to you is, I'm sure people you probably have done this, that if you stroke different digits, synchronized or asynchronized, what is the results?
The spatial discrepancy also can break down the illusion. So if, for example, the real hand is stroked on one finger or on the arm, and the fake arm is stroked on the finger, that breaks down the illusion. So any kind of discrepancy between the senses would reduce the illusion. And the bigger the discrepancy, the bigger the effect.
So if
Okay, I'm sorry, we're going to have to move on. We have only 30 seconds left. Two quick questions over here.
It is also for Dr. Sham. Can the causal inference model can apply to the various phenomena of body ownership, such as ownership of the third arm, ownership of the double body, or ownership of the body in the third person perspective? Thank you.
We have started working on the out-of-body experience phenomena. I think we haven't done it quantitatively yet, but qualitatively, it should apply to the third-person scenarios where, for example, you see your own body in front of you. Anytime that there's enough congruency between the senses to cause the integration or the inference of a common cause, this illusion should occur, and that applies to the entire body as opposed to just the rubber hand. So my prediction is that even though we haven't done it quantitatively, it would apply to those situations. The three-arm we have not done, and I haven't thought about it, but I would be interested in discussing it with you after the session.
And last question.
So I had asked earlier about the subjective sense of time as the level of anesthesia increases. I wonder if you have if you've done any experiments that correlate that with the physiological measures that you have and the rate of drug level.
When we talk about subjective measures, that's somewhat difficult to do in a rat.
I mean, obviously not a rat experiment. I mean, have you done human experiments with like an EEG?
So there is actually an ongoing study, and part of it has been published from our laboratory, which is conducted by Dr. Zirui Huang, and he is interested in the different time scales that associated with processing different types of information, such as syllables, words, sentences are processed at different time scales. And one of the things he found, and this was recently published in a Journal of Neuroscience, that time scales were differentially affected under anesthesia, and it appeared that the time scales were extended. And so you can read more about that. This is an fMRI study in human volunteers with propofol as anesthetic.
And speaking of time, unfortunately, we're out of it. I'd like to thank our wonderful lecturers, also wonderful audience, great questions. Thank you very much.